Could you please introduce yourself and your background?
So my name is Blažej Segelski. I have been a worker, and right now I'm a master's student in the group for virus biophysics at BMC. I have been working with the group for past 3.5 years already, and I have been involved mainly in working with microscopy, detecting the viral infection, and working with a system that is supposed to detect the infection of the viruses from a single genome all the way to something called replication compartments.
Okay, interesting. And how did you get into that?
It all started basically with me choosing my program, my path through academia, and I was always interested in working with viruses. And since Lund is not very big on research on viruses, there was not so many choices regarding the groups, but while choosing a group for my bachelor thesis, I stumbled upon Alex. Alex is my supervisor, and he's been working on this like interface between physics and biology, which is basically combining the physical aspects into modeling like living processes. And Alex has been working on herpesvirus. I came into contact with him, and this is where the journey began, basically. We've been working together ever since. I've been working on various projects on viruses, from antiviral peptides all the way, as said, to creating this new technology that is supposed to detect the viral infection.
Yeah, I've understood that you're working on both something that is very exciting, but also that you're creating some very interesting, unique insights. So it seems to work really well in your team, and it seems to work really well with Alex as well. He for sure seemed very happy to work with you. I'd like to capture a little bit about how it was for you to get started working with Cytely. So before Cytely, if I understand it correctly, there was a lot of manual collection of these cells that you wanted to find the cells that were both transfected and infected, but that was of course very hard to do manually. So could you maybe share a little bit about the pre-Cytely state?
Yeah.
Let's say.
Maybe I will start by introducing the system. So the system basically works in a way that we have a bacterial operon system. It contains a tetracycline repressor protein, which is supposed to be expressed in the cell. And then we have a tetracycline oppressor system that is introduced into the virus. How the system is supposed to work is that when the virus infects and the protein is expressed in the cell, this protein will bind to the operator's sequence on the virus. And this should like lighten up the single genomes, and how like when the virus is replicating, we should see where the virus genomes are. So this is how the system should work. And basically, in our system, we have been transfecting a plasmid containing the tetracycline repressor protein into the cell. And this system is generally inherently not so efficient, because transfection itself has probably around 20 to 50% efficiency. Viruses themselves, especially when they're infecting with very small amounts of viruses, not every single cell will be infected. So just by this fact alone, it's very hard to find those single infection events happening. And precisely how our workflow would look like is basically we would transfect the cells, then we would wait for 24 to 48 hours, and at that point we would probably infect the cells and then wait for X amount of hours to image them. And the process on the microscopy side, which is probably the most interesting for Cytely, is basically like that. We would sit by the microscope, we would try to find cells that look somewhat interesting. What was interesting to us is that if the cell is expressing good amounts of the fluorescent protein, the TET-FP protein, And then the search would begin. We would go from every single cell that we would have found on the dish, and we would check it manually on different confocal planes, whether there is something interesting. And this was especially annoying on the early time points, because on the early time points in the infection, you see just single genome, and single genome is visible only on single plane. So you would have to find a cell that is interesting. You would have to scan it all the way through, find something interesting, image it, and then move to another one. And this process would usually take us around 4 to 6 hours for one for one like single experiment. And thinking that we would have to make around 20 of those experiments and probably repeat some of them, it would be a very time-consuming process, especially that we'd have to also add to this image analysis and so on and so on. So everything together would be just very time-consuming.
And so what was in that pre-Cytely state, when you're spending 4 to 6 hours per experiment, do I understand it correctly that you are actually you are manually trying to find the interesting cells to image while standing at the microscope?
Exactly.
And so there, if I understand it correctly, and please correct me if I'm wrong here, but it also sounds like you are inherently with that process, and obviously it's not your fault, that's how it just works, but there's some level of human bias built into.
Oh, definitely. That's a very good point, actually. Yes, you would just be sitting and staring at the microscope for many hours, and of course, if you are sitting for so many hours, you kind of want to find something. And what this is producing is that sometimes you see things that you want to see, and it's not very objective way of doing that. Definitely there will be a very huge bias, especially when you later go and start to analyzing the images, and you see that, well, the images maybe not looking that good, and then you kind of try to force some kind of perspective into them. You just try to see things.
It's a bit like, when we're doing cloud spotting, when you're looking at the cloud trying to find animals or whatever, it just kind of showcases how we as humans, we are maybe too good at seeing patterns. We are very good at spotting patterns, and sometimes we see patterns where there are not patterns. And I can really imagine that if you're also facing pressure from having to have valuable data, and you're standing for 4 to 6 hours at a microscope, and I can imagine the different forces at play that might lead you to seeing patterns where there are no patterns. Okay, yeah. Thanks for sharing your experience there. And so, just for me to understand, what is the reason that way of working was the way of working before Cytely, and why wasn't that changed? How come the way of working hasn't changed before using Cytely? Because it sounds to me that it's with that inherent bias being built in the process, it just sounds to me that from a process standpoint, this could have been spotted earlier. And I'm in no way criticizing you or your team or I'm just curious, what do you think is the reason why this has been the status quo? Because I understand that also this is a status quo for quite many researchers, and tell me if you think I'm correct or off track here.
No, I think that you are absolutely on point. When it comes to my group, I think the main there has been taken steps to reduce this bias. Well, inherently, this is how you work with microscopy. So you just need to sit and take images and try to find something that is interesting to you. However, there is no objective way of doing that. There's always some kind of subjective level to what you are imaging and what you're analyzing later. So I think this is just a general concept in microscopy at this point, at least, when you don't have any automated and objective systems to employ. In our group, we have been starting through just looking manually and taking image of things that were looking interesting, because this is basically how our what our goal was to see the positive events, image them, and analyze them. And in the very beginning, we have been working with a very subjective system, which was basically, I said, look, image, analyze. Later, we tried to make it a little bit more objective, and that's why we employed a script for ImageJ. And this script is obviously a little bit more objective, since okay, you already have like image certain situations, but the script is the level, like it employs one more level of analysis, where it is more objective because it employs thresholding, and thresholding on the image based on the intensity of what we are imaging is more objective than our eyes.
Yeah,
So
that makes a lot of sense.
exactly, but this is the only level of automation and objectiveness that we could have added up to our experimentation. We couldn't, with the technology that was available, this was everything that we could have done at that point.
Yeah.
That is until we met Cytely, because Cytely changed a lot of things, how we view not only our data, but also how we approach imaging.
Automating scripts in Fiji can be quite complicated. I know that a lot of people just try to stay away from it because it's difficult. Was it you who did that automation?
It wasn't me. I was working together with Ramon. He's our physicist, and he had some background in programming, not a huge one, but together we basically worked. He first developed the script for another experiment, which was on viral capsids, which was done together with another doctoral student at our group. And later together we have been trying to redevelop the script into this new experimentation, because as you say, like Fiji scripts, they're quite tricky. There is a lot of we had a lot of discussions because there was many different ways that you can approach the problem, and yes, some of things you just is a little bit restrictive in doing, and we had a lot of discussions how we approach the problems, and it definitely wasn't an easy way to make the script.
Okay, so we talked a little bit about the status before Cytely, and we also talked about the reason for that being, let's say, the status quo. And I'm really curious now to hear, so when did you start using Cytely, and when did you notice, let's say, a significant impact in your work?
So it actually hasn't been that long since I've been working with Cytely. It's just been a couple of months now. I think 3-4 months since we started talking about it. But actually working with it, I would say it was around a month or two. And the results were quite immediate, I would say. Directly when we started working with Cytely, and like I got more fluent in using the software, which also wasn't that hard because software is very simple to use. After that, like the results were quite quick and easy to spot. Like it was much bigger difference compared to what we have been doing before that.
Okay, and so when you say that it was easy to use, how do you think that it differs compared to like what you have done in the past?
I would say that like working on microscopy and developing your own protocols of working with microscope is a very steep climb, especially like if you come from bachelor master level and suddenly you are starting to work with confocal microscopy for the first time in your life. From just learning what confocal microscopy is to developing your own protocols that are supposed to work quite well and be objective in what you are doing. This is a very steep climb for a person, and especially that confocal microscopy is not the center of your work. It's just a side tool that you are using to get your data after you have already done quite a lot of things before that. It's a very steep climb, and it differs a lot with Cytely because Like, I remember you had one very good line during one of your events that Cytely is just basically making this very accessible for everyone. And that's actually a very good way to summarize it. It's very, very accessible since you just plug in things into the microscope. Of course, you have still some of the user input in terms of like intensities, gain, and so on. But other than that, it's much more streamlined, I would say.
Okay, I understand. And so it sounds like Cytely has given you a shortcut in some way, but in a positive way, of course. And so, so from your perspective, what are the hurdles or the challenges that Cytely allows you to like not go through? Let's say. Do you understand what I mean?
Yeah, I understand what you mean. I would say that the biggest difference is just obtaining huge amounts of information in very short span of time. And it's also something that doesn't require me to sit by the instrument for long hours.
So, and that takes us back to the sense of how it was before versus now. So before, if I remember correctly, you spent like 4 to 6 hours per experiment, and then in terms of data, like how much data did you get out of those 4 to 6 hours of experiments?
That's a huge difference, actually. That's a huge difference. So before that, we could count on maybe, let's say, 5 to 20 data points per experiment. That was absolutely max that we could get out of it before the experiment would be rendered useless because of photo bleaching and other effects. 5 to 20 data points. With Cytely, suddenly you have access to thousands of data points, and this really shifted our perception of what we were doing. It changed the way that we approached the experiments, and it really allowed us to do things we weren't really able to do before.
Would you say it's a little bit of a leading question, but like in terms of magnitudes, is this a 10x improvement, 100x, 1000x? What are we talking about?
It's hard to put it in numbers, especially that I would say that it's definitely 100, even 1000x improvement, since, like I said, in terms of data points, definitely it's 1000x.
Yeah.
In terms of how we view the data, it's hard to put in numbers because I think the insight that we got just in the recent time was very invaluable, and we wouldn't be able to get it without Cytely. Like before that, it was just a notion. Right now, it's a straight fact.
Okay, could you just dive into that a little bit?
Just a very simple part of our experimentation, or how the experiment was supposed to work, was the process of transfection. And it has been a struggle for us for some time. We just couldn't really get the numbers there. I said it was like very hard to find something positive in transfected cells. And we never had an idea why is it happening. We were thinking, oh, maybe this is just a problem of transfection process itself. Maybe we have done something wrong. Maybe the viruses are wrong. Something was wrong to begin with, and we didn't know what. And for very long months, probably since the very beginning where I started working on the system, we've been struggling with that. We didn't just know what was the reason. So we were trying to optimize the transfection infection processes, and it was taking long months. And this all really changed the moment we got Cytely, because suddenly we were able to analyze the full sample. And by analyzing the full sample, we could very simply see how many cells were getting transfected and how many were getting infected and how many were both transfected and infected. And this really changed our perception, because this was the things that we have seen for the first time. And for the first time, we had the data on it to prove what was happening. And with Cytely, that's why we were able to check what is really happening. We were able to compare if we don't transfect the cells, how many of them will get infected? And once we introduce this step of transfection, how much will it change the statistics? And the difference was huge. Basically, based on that, we rendered the transfection process not good for us, not good for our application, because this system would make viruses unable to infect the cells. And this was like very huge step for us, because after that we know that transfection is not good. We need to have cells that are optimized for the system. We need a special cell line that is able to express our protein, and we just needed to eliminate the transfection from our experimentation. And this was something that we wouldn't be able to do it without Cytely, because as I said, for something like this, evaluating this process would require to see the full picture, and with Cytely we were able to see the full picture.
Okay, and like practically, how did that happen? I'm really curious as to like what was this Eureka moment, and what is the, let's say, unique mechanism with Cytely that made that happen?
I would say that was the Cytely demo that actually was the Eureka moment. Okay. Because suddenly, like we were just trying to test the Cytely platform with Carl, and suddenly we are sitting there with like me, my supervisor, and Carl, and suddenly you see like, oh, what is this? Like, we never seen it before. Like, why is there some few cells that are positive in both? And it was like the moment that we started to wonder, like, oh, maybe something is actually wrong with our system. And as I said, this was the first moment we were using Cytely for our purposes. And the very first moment we have noticed that, we see this for the first time. We need to analyze it and see like what we can do better. And this was the moment for us, actually.
Wow, that sounds super impactful.
Yeah.
Is this something that we could actually like look at right now?
Yeah, actually, I prepared a very quick presentation because I didn't want it to like go through specific experiments. So I prepared just a slide for you to see how it mainly looked like.
Yeah.
So this is how it looked during one of the first tests that we had with Cytely. We were basically able to collect the cells that were interesting to us and then compare them. So what we see with Alexa 488 is the cells that are transfected, and with Alexa 555 is the cells that are infected. And if we had cells that are being both transfected and infected, we would see a straight line going like perpendicular, but we don't see this. We basically see like a cluttering of cells in like specific groups. So groups that are just transfected and groups that are just infected. We didn't see both. And this was like the main changing moment for us, especially that we were able to see it directly with Cytely. I mean, we got this very first second, we just made an image, and it directly rose a question like, why is it happening? And after that, we got a hypothesis that, transfection is, inherently inefficient process. And second hypothesis that we had is that the transfection is basically a mimicking of viral infection process. It's coating of a DNA in a lipid particle, and this particle introduces a foreign DNA to a cell. So the hypothesis was that this will activate an antiviral state in the cell, and after the transfection happened, we wouldn't be able to infect with the cell since the antiviral state was already introduced to the cell. And then we were able to test it, and we performed 3 parallel experiments. We did the cells that were transfected and infected. This was the first experiment that we have done. Then we tried to do it with untransfected cells, so cells that didn't have this foreign DNA introduced. And we see directly that all the cells are quite positive in both, so there was no grouping. And once we also tried it with cells that normally express TTR protein inside of them, and we see that they instead start to group in a single line. So basically, they both transfected and infected. It was not transfected, but it has a protein already. So the protein itself doesn't render the cells unable to be infected. And this is basically the part where we started to understand it, that we cannot transfect our cells if we want to see infection events. And this was a big moment in our group, since the transfection has been basically a cornerstone of our work. We always work with the transfection, and we always thought that this would be the best process for us. But after that, it changed our perception, and now we know that we cannot do that.
Wow. Yeah, that is really, really cool. Because that's a like from my perspective, this looks like the transformative understanding came from like almost like emergent exploration of your data. So like you were, if I understand it correctly, being able to get the population-wide view of the sample allowed you to spot a pattern that signaled that maybe we're actually not even doing the experiment in the right way. And so based on seeing that initial signal, you acted on that learning, let's say, you tested a new approach, and then you could immediately see that your hypothesis, let's say, was correct in that the experiment wasn't correctly set up. And do I understand these steps correctly?
Yeah, it's correct. It's correct.
Okay. And so the charts that you showed us, that's that's from Cytely, right?
That's from Cytely, yes.
How was the experience for you in using Cytely in this way to further your understanding? So I'm curious, like, both from the perspective of like intuition, is this something that was natural, you know, and also from the perspective of is this a potentially a new way of working? And I'm sorry for the leading questions, but it kind of just I'm trying to paint the picture of what I'm asking, if that makes sense.
Yeah, I would say I do understand your question, and I think that with Cytely it's very straightforward, since I mean, this is something that you would do normally just to check like how do fluorophores distribute in the population. So it was very straightforward, but and very quick to begin with, because you get this data directly. And so you can directly go and check what is actually happening in the sample itself. So I would say that it's the logic behind this is very straightforward, but the way you can obtain this data so quickly and directly when you're working with the sample is completely new.
And when you say directly, what do you mean?
I mean, you just like by sitting by the microscope, already in that moment, you can see what is happening. And normally this wouldn't be possible because you need to take your data with you and then you would analyze it. So by the time you have these numbers in front of you, it's already too late. Like you cannot go and check every single cell again. So I think this is like the big difference here.
I understand. Yeah, so we're almost talking about two different philosophies, where you have, on the one hand, you have an image-driven philosophy, let's say, where you want to find a nice image, a nice cell to image, and then you want to study that. Whereas on the other hand, we're talking about more of a data-driven philosophy, where you first want to understand your sample, from almost like index it, and then based on that you want to go deeper based on like data-driven parameters. Am I understanding this correctly?
Definitely. I would also say, I would actually put it differently. I would say that in terms of data, I think you either work in the present or in the past, right? In this sense. Like if you're working with Cytely, you're working in the present. You see what is happening, you're acting on what you see, and you can either change your approach or improve it directly. Meanwhile, when you're working like with our old ways, you're acting on the past. You see what have happened and you don't have any influence over it. Like it's not very good way of working if you're working on the past, like you don't have influence over it anymore.
That's a fantastic statement. You're like with Cytely, you're working with data in the present and versus the past. Yeah, amazing. Yeah, thanks a lot. Okay, so it's been. I'm really happy about where we're like diving into your different experiences, and it's way off track from the questions that I prepared, but I have prepared some questions.
Yeah.
And we already touched a little bit on some of these. But if we can, maybe if you can just concretely quantify how has Cytely changed the way you work, and what kind of improvements have you seen?
If it has to be quantifiable, I would say that I am able to do right now the work that I was doing throughout maybe some months, like 2-3 months, I am able to do within a week or two. Not counting even data analysis, because data analysis would take even longer. Meanwhile, data analysis here is prepared for you and put on a plate in front of you, so it's much quicker.
It sounds like there should be a compounding effect there, right? Because if you do your experiments faster, then I would assume that you're also you're learning faster because your iteration loop is shorter, and so if you learn faster, then you also make better decisions earlier. So it should be like
it's definitely a curve, I would say. Even the whole fact that something that I was working on trying to troubleshoot for 3 years, I troubleshoot within first few moments working with Cytely is quite remarkable, I would say.
Yeah, I agree. I'm so happy you got that result, by the way. And not just because, of course, it's fun that it's with Cytely, but also it really sounds like it mattered a lot to you, and that just makes me happy.
Definitely.
We've heard a little bit about from our other users that it's easier to work with the data, because it's accessible in a different way. Do you have any experiences in that domain in terms of like being able to access your data and so on?
Definitely some. So basically, I would say one of the very things that becomes very apparent with Cytely is that you can work anywhere, and you don't need to carry a very heavy hard drive anywhere with you. And hard drive always can something happen to hard drive, but it cannot really happen to the data if it's stored in the cloud, right? So that's definitely a difference here. It's much more accessible. You can work anywhere where you want with it. It's already a big difference because I've been traveling throughout Christmas right now, and I could have accessed my data and work with it when I had some time anywhere. It didn't matter if I had the hardware with me or not.
Oh, and that wasn't the case before?
No, definitely not.
How come?
Because, it's very heavy data. It's lots of images, and I wouldn't just be able to do it before, because maybe I could, but it would be much more of a struggle than it is right now. Right now I can just log in anywhere and just work with the data directly.
And what about like, because a lot of universities obviously have great infrastructure for saving data and so on, and I'm sure that Lund University also has some alternatives, and so it was in terms of, so how come that it was more convenient for you to use Cytely compared to using existing, data storage infrastructure, let's say?
I think it mostly goes down to our group that we had issues before with online data storage. That's why we moved. We had some parts of the data disappearing after there has been issues with data storage, and after that we decided to store everything on hard drives instead. And I think it's mostly like our preference in the group that we prefer to work with hard drives. But of course, this is making the access much more difficult, especially that we are multiple people in the group, and the hard drive is one. You cannot, not everyone can have it at once. Meanwhile, the data is right now accessible to everyone.
Yeah, that makes sense. And and it's obviously an aspect that we really enjoy helping researchers with, because it doesn't make sense that each research group needs to build their entire data storage infrastructure.
I'm a little bit curious about, so when you've collected the data, of course, you need to present it to people and to show it to your supervisor, for example, and so on. I'm just curious, has this changed using Cytely versus how you did it before, or is it?
I would say that definitely, right now it's much easier to convince my supervisor to my ideas, because I can show it directly. It's just a fact-based way of showing it. It's just to log into Cytely platform, just show what I have done, and there is no other way to manipulate the data. You just see the straight out facts. So it's somehow it's changed how you present the data. It's much quicker, much easier to show it and much more objective way of presenting it. Because you just work with a full data set, and you can manipulate it directly in front of the person that you're presenting this to.
What kind of tools did you use before?
It will be just spreadsheets. You extract the data yourself, throw the data into a spreadsheet and then try to present in some kind of a graph form. Right now, we just don't, like with Cytely, we don't have to do it. It's right in front of us. So we see how populations are divided. You don't need to make the populations ourselves. I know that we could do it with R, but this is just a struggle. Here you get statistics in front of you. You don't need to have multiple programs working with. And as I said, a much more streamlined way of working. You have everything accessible to you in one place.
So if I understand it correctly, previously it was not just that you spent hours with the microscope, it's also that there was this like huge post-processing manual labor with like thousands of Excel sheets here and
Exactly.
hard drives, and okay, it sounds like it.
Yeah, I mean, it was so I can actually draw a very quick glimpse into what I've been doing. So
yeah, please.
After the imaging, usually what will be happening, you will have to go through every single image that you have done. So even if it's 30 to 40 images per each sample, it will be, and we have a couple of samples per experiment, it will be still quite a lot of things to look through. And you need to go through every single image to see, for example, the threshold. So what is the intensity on the image, and you need to apply a threshold to that. And
wait, so sorry if sorry for interrupting, but just for me to understand, like, so you had to look at each image by yourself and then
yes.
apply a script on it, or like
even before you apply a script, because script is working on the script needs to have a certain threshold. So it is a threshold of intensity on the image. So for the script to calculate it, you need to tell the script that everything below this threshold is uninteresting to us, and everything above that threshold is interesting to us. So you would need to go through every single image, even before you apply the threshold to the script. You will need to go manually through each image and choose the threshold for the image. And here is another problem, because normally you would have to do some you would need to find something that is optimal for a couple of images and not applied for a single image. And you would need to think like what would be better, where I can get more data by applying this single threshold to all of the images at once. And it would just be taking hours upon hours after you already imaged it. And only then you would use a script. And after you use the script, you need to pull everything into the spreadsheet, and then you finally can see your data. So sometimes, like when you did an experiment, you would see the data only a week or two afterwards.
Oh my God, that sounds so painful. I'm really sorry I had to experience that, Blase.
I'm sorry for myself as well.
Oh, see, it almost sounds like 80% or 90% of the time spent by researchers in that situation is not on like generating insights. It's just like plumbing.
Exactly. Yes, it's very manual labor that doesn't create any new insights, doesn't create new understandings. It's just unnecessary, I would say.
It's such a waste of so good brains.
Exactly. And just think about it, that it's not only me, but it's thousands of researchers that are working on the microscope every single day.
Yeah, exactly. And so connecting to that, let's imagine for a second that every researcher in the world has access to Cytely. What do you think would be the impact on our civilization?
That's a very far-fetched question, actually. But I would say that with just being able to see the data quicker, I would say that maybe it could make a progress quicker. Plus, objectiveness of this could eliminate some insights that are wrong to begin with. Mm-hmm. Like as said, we also had assumption from the very beginning, and we were trying to work around this assumption and trying to make the assumption work, not trying to make the experiment work.
Okay, and so from your perspective, why is it important that that we have? And it sounds like a silly question, but I'm just trying to understand different perspectives here. So for you, why is the objectivity in the data important?
I mean, that's quite obvious. Why is it important? Like, you need a objective approach to, well, basically, even if you want to adjust your data, right? If you are subjective, you are just applying this to a single experiment, single sample, and it's not applicable, won't be applicable to the rest, because you're just trying to apply it to a single single solution to a single problem, not looking at the whole picture of it. With objectivity, you can basically apply a single solution to everything. As for example, like when we're working with infection, right, and transfection. Now we have an objective data that says, well, you cannot apply transfection simultaneously with the infection because this will render infection impossible. Without it, if you were trying to approach the data subjectively, we would try to find some issues with, maybe in this experiment, the viruses were slightly more mutated and they didn't have the sequence. That's why we just couldn't spot them. Or maybe, there wasn't just enough expression. Maybe we did something wrong with the experiment. It's just not a good way to approach it.
Yeah, no, that makes sense. And as I said, it's a silly question, but it's always nice to hear the perspective. As there are many, believe it or not, there are many different perspectives on this point. So I just wanted to get your thoughts on it. But so if we say from 1 to 10, so let's make this a bit quantitative. From 1 to 10, how much do you want to go back to your old way of working?
That's a very good question. I would say I would go by 1 probably. I definitely don't want to go back into what I've been doing before.
Yeah. So 0 if it's allowed, right?
Yeah, 0 if it's allowed. I would even forget about that way of approaching my problem.
Yeah, I can imagine that, especially based on what I've heard from you. So I understand. And from 1 to 10, how much do you want to keep using Cytely?
Definitely a 10.
And how can we make that into an 11?
How can you make it into the 11? I would say apply a zoom button on the population overview, please.
Okay, zoom button.
Yes.
Could you dive into a little bit more why you say that?
It might be a silly thing, but when I was working with Cytely, it's just like, there are certain things that come very automatically to you as a user that it would be nice to have. As for example, like even changing the name of experiment, right? You just sit by the microscope. It's like your 10th experiment of a day, because with Cytely you can actually do that. You can do 10 experiments a day. You sit by the experiment and you just mistype something, and then you cannot change it, right? So,
okay.
Just a small thing, but there's something like this just comes into mind. But zoom button is definitely needed. Like if you have 10,000 of cells and one will be at the level of 1,000 and the rest will be somewhere at around 100, everything like kind of clumps together, right? So it would be nice to just zoom into one population.
Okay, and so is this like a zoom in the chart?
Exactly in the chart, yes.
Okay, yeah, that's a brilliant suggestion. Yeah, thank you. Any other things you thought about?
The only thing left, I would say, is that when you actually pick a cell, like if you have this population, right? You have the window and you have a single dot. You maybe like want to choose this single dot, and you have a picture on the bottom of the cell. You would like to see the cell, exactly this cell in the file. You want to like take the raw file of this cell. You still cannot do it personally. You need to like kind of look through all the pictures, and if you take in 100 pictures, it's just very difficult to find as well.
Yeah.
But that's just very few things, I would say.
Yeah, well, super good suggestions. And whenever you think about these things, don't hesitate to use the chat on the platform. We all see the messages, and like these types of user-originated suggestions, they tend to get into the product quite fast. So you are not only helping us, you're also helping yourself.
Yeah, exactly. It was both ways, right?
Yeah. Okay, so we're almost done here, and thank you so much for the candidness. It's been really, really nice to hear your experiences. Just wanted to ask, so imagine that there's a blasé somewhere else who is spending 4 to 6 hours per experiment, getting very little data, and they're watching this right now. What would be your suggestion in terms of taking action on using Cytely, for instance?
Well, basically, if you want to get any kind of if you're working with samples that require huge amounts of data, then definitely Cytely is the way to go. I just don't see a better way of approaching this right now with all the tools that I have been going through during my work. I was trying to find some tools to streamline the process. There's basically nothing that would give this possibility to do it that easily and that straightforward at the current moment. There's basically no other way of doing this.
That's a very clear recommendation. Thank you. Okay, Kalle, do you have anything you want to Ed?
I was just a little bit curious, perhaps, about we have the data-driven acquisition, where you select a certain population and then use that to image cells at perhaps a higher magnification. I was just curious, what has your experiences with that been? Have you used it much, or?
I've used it some. I actually developed a new protocol, like my own protocol of working with it to make this process a little bit easier. At this point, I haven't been working much because it kind of mirrors what I have been doing as well. It's just easier this time because I know, like before, how I would approach like imaging first at lower magnification, then approaching a higher magnification. Would be just I just see the cell, I save the location of it, and after that, like after I already went through 20, 30 cells, I would change the objective and then come back to the certain locations to image them. This is very similar with Cytely, but this time like we can have just more. It's kind of the same pattern that you see overall with Cytely. It's in some aspects it's the same, but you just get more of it. Much more of it. So like I said, before I could save maybe, I said, 20, 30 locations before I started imaging them. Right now I can do it to 100 cells and just press one button and then it will do it automatically for me. Before I would still save the locations, I will automatically go to the location, but I would have to image it manually.
And what about bias in that particular process?
I would say that the bias is somewhat smaller because when I was doing this myself, it would be just me seeing something that is interesting, saving location, going farther, and then coming back on the higher magnification. Right now, it's more driven by the facts because I'm approaching this by intensity, I'm approaching this by the cell size and so on. There's still some subjectiveness to it because in our way of work, there is some things that you cannot be objective about. It's like there's absolutely no tool yet that can also do it. It's basically distinguishing cells that do have infection going on because you just cannot see it objectively yet. So there's no other way of doing this. So there's still some subjectiveness left, but it's quicker. It's still has some objectiveness to it based on analysis of like cellular characteristics like size, circularity, and so on. Something that we haven't been doing before.
Place, thank you so much. This was really amazing to be a part of, and what you and your team are doing is tremendously important for humankind from where I sit, at least. And so thank you for your work, and thank you for making it so rewarding for us to build Cytely for experts just like you.
Likewise, I mean, without you, I would definitely not be able to do what I'm doing right now. So it goes both ways. Thank you so much. It was a pleasure working with you and giving you some of my feedback.
Yeah. All right, and let's keep the channel open. So this is not just like a one-time thing. We want to keep this kind of conversation going with all of our users and customers, and really, like, you're making a huge impact with your experiences and with your sharing. So thank you.
Thank you. Like us.